Welcome back. Today we're going to be talking about the 4 different types of macromolecules. And remember that macromolecules are made from adding together repeating subunits, and there are 4 types. So we have carbohydrates, proteins, nucleic acids, and lipids. And you might see that I have like a kind of star here about the lipids. With lipids, it doesn't work quite as well, but we're going to just go with the whole macromolecules are polymers that are made from monomers. So let's talk about these 4 different types. Carbohydrates you've probably heard before. The monomer of a carbohydrate is called a monosaccharide. You can kind of see what it looks like right here. This is glucose. We're going to talk a lot about glucose in the next chapter, so take a quick look at it. You are not responsible for being able to reproduce this structure, but you will need to know that glucose has 6 carbons and it forms a ring. The polymer of a carbohydrate is called a polysaccharide. So remember, here are our mono, meaning one, and poly, meaning many. Monosaccharide and polysaccharide, or carbohydrate. That's the polymer of carbohydrates. When we put them together in a dehydration reaction, we form a covalent bond between them. If we were to take maltose and go the other way, add water in and go the opposite direction, we would get a hydrolysis reaction, and that's what you get when you do digestion and you break down more complex sugars into simple sugars. Carbohydrates tend to form really long, like thousands and thousands of monomers long monosaccharides. You can see them here. Each monosaccharide is held together with a covalent bond, and these make these really, really long, kind of branched or sometimes unbranched chains that make up the carbohydrates. Now there's a couple of different functions for carbohydrates. In terms of a monosaccharide, those are immediate energy. When you think of glucose, that's a monosaccharide, and it's going to be very easy for us to break down and break apart and use it to fuel our cells. And you kind of know this already because if you've ever eaten a candy bar, you know that you're going to get an immediate kind of sugar rush and a ton of energy from that. Now we also use carbohydrates for medium-term energy storage. So not immediate source, but we store it for a little while. Examples of medium storage include starch, which is what plants use to store energy, and glycogen, which is what we and other animals use to store energy. All right, our next type of macromolecule are proteins. The monomer here is the amino acid. You can see what it looks like over here. And amino acids, when you make a macromolecule of protein, are held together again by covalent bonds. In the case of proteins, we call it a peptide bond. It's just a special name for the good old covalent bond. And so you can have a peptide, which is a few amino acids, or a polypeptide, which is many amino acids, or you could have a protein, which is thousands and thousands of amino acids. Now there are 20 different types of amino acids, and the big difference between them is this R group here. So all of the amino acids share the same structure, but the R group might be different. Proteins are so important in the body, it's almost impossible to come up with just one function of what they do. You find them everywhere. So I have a couple things that I wanted to go over. So one thing that's really important is that we have receptors that are embedded into our cell membranes, and those receptors help to receive signals from outside the cell and tell the cell what to do. And those receptors are protein. They're made out of protein. Proteins make up antibodies and clotting proteins that we might find in the plasma of our blood. Proteins also make up the matrix of some types of connective tissue, and they're really important in maintaining our water balance as well as our pH in our body. But really, the most important thing that we do with proteins is we make enzymes out of them, and enzymes are essential to carry out the reactions of life. Remember a few slides ago, I was saying that we need enzymes anytime we add together monomers or break down polymers. Enzymes are required for performing all of the reactions, either adding or breaking down, that make life possible. And again, enzymes are made out of protein. There are several levels of protein structure, and the proper folding of the protein is very important because it gives it the proper shape. And the shape is what determines whether or not this protein can do its job, that can make the reaction happen. When you denature a protein, you cause it to fall apart. You cause it to not have the proper shape anymore. And there's a couple of ways you can denature something. You can denature it because of the heat. You can use chemicals, or you could use either very high or very low pH, and all of those are going to cause the protein to denature. So let's look at these protein structure levels. So remember I told you that there were 20 different possible amino acids. And if you think about it, each little bead on here is a different amino acid. So there's lots and lots and lots of different combinations of those 20 different amino acids. And which ones are used and how many are used is our primary structure. And remember, each one of these is held together by a special kind of bond called a peptide bond. They're all covalently bonded together. So that is our primary structure. The secondary structure is sort of a local folded structure. So if you have a long polypeptide, right, a long protein, little parts of it are going to start to fold. So for example, you can have it and then all of a sudden this part folds up a little bit like that. And this part might actually fold up like that. That's secondary structure. So it's not the whole thing folded up, but it's just this area and this area are folded up. And it folds into two different types of structures. An alpha helix, which almost looks like an old school telephone cord, right? And a beta pleated sheet. And that looks kind of like a paper fan or something like that. And the reason these guys are being held together is our good old friend hydrogen bonds. So hydrogen bonds hold together amino acids from one part of the chain with an amino acid from another part of the chain and causes the chain to form these sort of localized foldings. That's our secondary structure. Our tertiary structure is the overall three-dimensional structure of the polypeptide or the protein. And you can see here, there's an alpha helix right there. Do you see how it goes round and round and round? And I don't really see any beta. Oh, here's a beta pleated sheet right here. Do you see how it goes back and forth? Right? So it's got the secondary structures in it. We know that the primary structure is just whatever the amino acids are that are making up this chain. But the fact that it's completely folded up now, the entire thing is forming a three-dimensional structure, that's our tertiary structure. Tertiary structure is really important because it's the minimum amount, that's a terrible star. All right, it's the minimum amount of structure that you need for this protein to be functional. Remember how I told you that the shape of the protein is really important for its function? Remember, structure, function. So in order for the protein to function properly, it has to have the proper structure, the proper shape. And so this tertiary structure folds the whole thing up. There's one more level of structure. So what does that mean? It basically means when you have more than one polypeptide. So you can see you actually have 1, 2, 3, 4 different polypeptides in here that are making up our quaternary structure. So it's 2 or more of these things. This is an example of hemoglobin, which carries oxygen in your blood. It has 4 different protein chains that come together in quaternary structure. Not every protein has quaternary structure. Sometimes just one protein strand is enough to get the job done. So primary structure, again, are held together by covalent bonds. In proteins, we call these peptide bonds. And remember, these are pretty strong bonds. Secondary structure is held together by hydrogen bonds. And remember, we talked about these as transient attractions. So they can be pretty easily pulled apart or broken. Our tertiary structure is also held together partly by hydrogen bonds, but also some other types of attraction. And quaternary structure is held together pretty much the same way. So when you denature a protein, remember that causes a protein to unfold. It's because we're breaking the more transient and weaker bonds. So we're going to get rid of quaternary structure. We're going to get rid of tertiary structure, so we can no longer hold a three-dimensional shape. We're even going to get rid of those hydrogen bonds that are holding our secondary structure together. So all of these are what we get rid of when we denature proteins, either by heating them up, exposing them to extremes of pH, or other chemicals. The primary structure is usually maintained unless you have some pretty significant enzyme activity. Okay, let's talk about our third type of macromolecule, nucleic acids. So the monomer of a nucleic acid is a nucleotide. A nucleotide is shown over here. It's made out of a phosphate, a sugar, and a base. If you've taken biology before, you might remember the A, T, C, G. Those are the nucleotides. There's 4 of them. The functions of nucleic acids is that they store the genetic code in your DNA. We use RNA, which is also a nucleic acid, to make proteins. And ATP is what we use for cellular energy, is also a type of nucleic acid. ATP is used for cellular energy. ATP stands for adenosine triphosphate, and instead of having one phosphate, it actually has 3 phosphates that are held together with covalent bonds. Now, because each of the phosphates is negatively charged, they want to get away from each other. They are not attracted to one another. And so when we put covalent bonds between them, those bonds have to be really, really strong. And so we can store quite a bit of energy in the second and especially the third bond. So we can use ATP as kind of a way to store energy. When we put the phosphate on, this bond right here, the third phosphate bond, is very high energy. When we break off that phosphate, the energy is released, and we can transfer it and use it somewhere in the cell. This results in something called ADP, adenosine diphosphate, because it has 2 phosphates. Then, when we get extra energy, we can kind of cram one of those phosphates back on the end of it, store energy in the third bond, and then we have another ATP, which can be used and broken down. So we go through this cycle of ATP to ADP, and then we recreate ATP again. Our fourth type of macromolecule is lipids. Now, the monomer here is a fatty acid. There are actually 3 different types of lipids. So triglyceride, which you'll see right here, has 3 fatty acid tails. And we use this for long-term energy storage. So for example, if you are eating a whole bunch of food and you don't use it all right away, we might store it in our body as triglycerides. This is another word for fat. We also have something called phospholipids. This is another type of lipid, and these are used to create the cell membrane. You can see that it only has 2 little fatty acid tails, and instead of having a third fatty acid tail, we actually have a phosphate on it. And the last type of lipids doesn't really fit with the other 2 because it looks totally different. It doesn't have fatty acids, and this would be steroids. So for example, this is the structure of cholesterol. Some of your hormones like estrogen and testosterone have a very similar structure to them, but you can see how they have these kind of odd shaped rings. So again, these are all lipids. That's the macromolecule, and the monomer is fatty acids. Depending on what type of lipid we're talking about, they have different functions in the body. Thanks for watching. See you in class.